Thank you very much indeed. I'd like to start by thanking the organisers for the opportunity to attend a superb meeting. We have already heard some wonderful talks and seen some beautiful science, and I'm grateful for the chance to present some of our own work. As a starting point, it's familiar to everyone in this audience that we have an immense load of microbiota in the lower intestine, tenfold more than our own cells, and they have cumulatively about 100-fold more genes than are in our genome. And it is quite clear from the previous talks and has been established in the last 10 years, and the people in this meeting have made some very important contributions to this understanding, that the commensal microbiota that don't actually have pathogenic effects nonetheless have profound effects on the host, especially the immune system, which is the subject of my talk, but very widely amongst many systems and can be seen as a super system. And the reason that we know this is through germ-free work, that we can keep animals germ-free in these flexible film isolators here, fed with sterile bedding, food and water through these transport drums that go into autoclaves and then transferred through a peracetic acid sprayed sleeve into the isolator. And the comparison between germ-free and colonized animals actually allows us to make this quantum leap in our understanding. And in the last 10 years, it also has been possible to combine this with strain combination experiments, either by caesarean section or, as shown in our laboratory, by axenic embryo transfer, in order to be able to compare different strains within the supersystem of the microbiota. And this indeed shows that there is powerful induction of the immune system as one of the important systems that's influenced by this, shown in these panels comparing germ-free black 6 mice with black 6 mice that have been colonized with intestinal bacteria but are pathogen-free. And you can see in the staining that there's a considerable upregulation of IgA, that there is increased size and number of germinal centers in the secondary lymphoid structures of the intestine, the Peyer's patches, also increased size of the isolated lymphoid follicles. And there are many other changes as well, illustrated here by increase in the CD4 T cell numbers in the intestine, just as an example. So if I start by summarizing some work that we have presented and published in the past, that there are low numbers of commensals sampled by intestinal dendritic cells, and these are capable of inducing IgA mucosal immunity, and the B cells thus induced travel through the lymph and then the blood home back to the mucosa and secrete IgA that heavily binds organisms within the lumen, that there is a kind of firewall through the mesenteric lymph nodes because the induction process happens locally here in the mucosa, and the dendritic cells can actually traverse to the mesenteric lymph nodes but don't, as a rule, penetrate further into the body, separating induction geographically in the mucosal immune system from the function, as I've shown, in recirculation. Innate function is clearly necessary to mop up small numbers of bacteria in dying dendritic cells and those that manage to translocate the epithelium. And we have also shown that the serum responses against commensals actually form a continuum when the innate function is defective. So the sorts of experiments that have been done, and it's been possible to keep germ-free animals now for almost 100 years, is as described this morning, that you can look sequentially after birth at colonization and the production of immunity. You can gavage organisms from pure culture and then look at the induction of immunity, or you can start with germ-free animals and simply add a colonized animal with the required defined flora to the same cage, and the germ-free animals will acquire that microbiota and look at the effects on the previously germ-free animals that then become colonized. I'm sorry, I'm having some difficulty with the pointer here. Okay, you'll just have to bear with the rather anemic effect. Okay, so you've heard about segmented filamentous bacteria, and our approach has actually been to simplify the microbiota as far as we possibly can. And we have generally used the altered Schädler flora as our defined flora. So we take germ-free animals and we deliberately colonize them from pure culture with these 8 organisms in order to precisely define what we have got and to eliminate other organisms from the microbiota so that the effects are reasonably clearly defined. And what you can see here, if we do the sort of experiment that we colonize germ-free animals and then look at the consequences, is that as the animals have become colonized, whether they're Black 6, Balb C, NIH Swiss, and we've done these with a variety of other different strains, 28 days after colonization, you see an increased population of regulatory T cells. When these are studied with the Helios stain, they have the characteristic of being Helios low and therefore putatively induced regulatory T cells. And this is a response that is specific to the mucosa. As you can see, if we study the variety of strains in the spleen, the mesenteric lymph nodes, or the Peyer's patches, there is no significant difference germ-free and after colonization. And the effects on, remember, these strains don't have SFB, segmented filamentous bacteria, so there are therefore modest effects on Th17 and Th1 cells. These are studied within the colonic mucosa. So what is the functional consequence of this within a benign intestinal microbiota? Well, we had the fortune to have smarter transgenic mice. These actually have an irrelevant T cell receptor transgenic specificity on CD4 cells to a component of lymphocytic choriomeningitis virus, but as I say, it is essentially an irrelevant specificity. But what we noticed was that a proportion of them have a rectal prolapse. And when we studied these germ-free and colonized, we found that they were unable to do this upregulation of regulatory T cells within the intestinal mucosa. So here is the Helios stain, and here is the wild type shown as a comparison, and the Helios low is absent following colonization in this strain. We therefore sought, and this work was done by a talented postdoc in my lab, Marcus Goiking, we sought to see whether or not we could transfer regulatory T cells and correct a defect that we had noticed, that if we colonized these germ-free smarter mice for 28 days and then looked at the proportion of IL-17 expressing cells, we found that now there was an increased proportion of these Th17 cells. And indeed, if we transferred in, before we did the colonization, regulatory T cells, we found that this response could be abrogated. Remember that these mice have this defined altered Schädler microbiota of 8 strains without SFB, and SFB itself can be seen, as the previous speaker said, closely attached to the epithelium, here actually spearing into the epithelium. So although it is unquestionably a commensal in mouse strains, it's not, in my view, an entirely benign commensal. And the point of these experiments is to show that SFB is an important part of the story, but certainly not the whole story, and that the immune regulation, the regulatory T cell responses underneath SFB are important, even in the face of what I have rather cheekily called a truly mutualistic microbiota. Okay. So another way that we have developed, and this has been done by another talented postdoc in the lab, Siggy Hapfelmeier, of looking at the immune responses in the mucosa to commensal organisms in this rather tightly defined way, has been not to take a germ-free mouse and simply colonize it, but to see whether we can go backwards. Now, of course, you can take antibiotics, but that is only going to reduce your microbial load by some orders of magnitude, and these will all recover quite quickly after you cease taking the antibiotics. So it isn't the same as going to a germ-free status. And after quite a lot of work, the idea was to be able to gavage with live bacteria and recover the germ-free status and then study immunity afterwards. So some of the criticisms of germ-free work are that the lymphoid structures, as I have shown you, are rather undeveloped. The mucus layer may be a bit different. There are a variety of other expression changes. And so looking functionally at the consequences of mucosal immunity before you've corrected all these changes may be subject to some criticism of interpretation. So the notion was that we should be able to uncouple priming of the intestinal immune system from colonization. And if we could do this, we should also be able to do something else, which was to prime the bacteria mucosally, prime the mice mucosally with live bacteria, recover the mice germ-free again, and keep on doing it. In other words, to be able to study mucosal immune memory to these organisms. And the way that we chose to address this was to create a strain of Escherichia coli that had auxotrophic deficiency for certain components that couldn't be supplied by the host, the mouse. And we chose initially diaminopymelic acid as a component of the cross-link of peptidoglycan. And when we did this at first, most of the experiments were reasonably successful, but a few mice, after we had gavaged them with this strain that was incapable of synthesizing diaminopymelic acid. Of course, you can grow it in culture simply by adding the diaminopymelic acid as an auxotrophic requirement to the rich medium. And so when we put it into mice, most of the mice became germ-free again, but a few actually started to secrete fecal organisms. And we looked at these in comparison with the same strain grown in the presence of diaminopymelic acid, and as you can see, the colony structure is rather different. And our interpretation of this is that they had managed to get around the requirement for diaminopymelic acid by altering their cross-link. Not to be deterred, we of course knew that D-alanine is different from the L-alanine that is used in mammals, and therefore Siggy deleted the two enzymes that are required as stereoisomerases for D-alanine, and so we had a triple auxotrophic mutant that we could grow quite easily in culture in the presence of diaminopymelic acid and D-alanine, and then gavage into mice. Of course, I wouldn't be telling you this story if it didn't work. And you can see in the black filled circles, if you give the wild-type parent strain to germ-free mice, there is over time a very nice and consistent colonization. If you do this, and the strain is HA107 with this triple auxotrophic mutation, then there are live bacteria within the feces, and then as one goes out to 48 hours, these bacteria disappear, even if we do amplification cultures in rich medium, and the mice are completely germ-free again after a while. And we can do this sequentially, and the mice consistently become germ-free. So the tool works, and the question is, does this have any effect on the mucosal immune system? And again, I wouldn't be telling you the story if it didn't. And you can see that compared with germ-free mice, and here the fluorescence is for IgA, at day 14, these mice that have been treated with the reversible colonization strain have a significant development of IgA, and in fact, this is just as good as the IgA that appears at day 14 if we colonize the mice with the altered Schädler flora, the irreversible colonization with the 8 defined organisms. So the next thing that we wanted to do was to look at the specificity of this response. There were some ideas in the literature that the IgA was just sort of immunological flypaper produced as a series of natural antibodies that didn't have a significant specificity to commensals, although it's always been acknowledged that it does to pathogens. And to address this, Emma Slack in the lab developed a flow cytometric-based assay. And I just need to show you this quickly so that you understand some of the downstream slides. And what this is allowing the antibody, in this case serum IgG, but of course most of the slides I shall show you are with intestinal IgA, to bind to the bacteria and cause a shift. And that shift, the extent of which depends on the dilution, in this case, of the serum or of the intestinal IgA. If we immunize the mouse with Enterobacter cloacae and then do the test with Enterobacter cloacae, then you see the shift. If it's immunized with Salmonella typhimurium, there's no shift when the test is with Enterobacter cloacae, but there's a nice shift with Salmonella typhimurium, and vice versa. In other words, this is a very nicely specific readout system. So the question is, can we see the specificity of readout with a reversible colonization in these mice? And the answer is we can. Each bar shows a series of dilutions. Of course, the amount of intestinal IgA is different in germ-free from colonized mice, so it's necessary to show that as a concentration on the x-axis, and on the y-axis is the shift as a fluorescence geometric mean. And you can see that after colonization with HA107 in mice that have become germ-free again, there's a very nice shift that is not seen to the ASF, which has no Escherichia coli within the 8 ASF organisms, nor in the black to germ-free controls. If we do the same thing with Salmonella typhimurium, as implied by the previous slide, there is no shift from the priming to HA107. So the priming itself is specific. Next thing is, do you actually need live bacteria to do this? Is it not enough just to put dead bacteria or dead bacterial components into the intestine to induce some sort of non-specific response? And the answer is no. You can see here, if we compare live and killed bacteria, so they've been killed either by heat treatment or by peracetic acid fixation, you can see that there is consistently a much better response to the live organisms, shown here with black 6 mice, also shown with Swiss Webster mice. The effects with killed organisms are always really rather weak. The next concept is, how do we respond in terms of our threshold to these organisms? How many organisms do we need in the intestine in order to be able to respond? This, of course, has been impossible to address before, because either we had to do gavages of mice that already had an intestinal flora, when the intestinal flora would actually compete out for space with anything that we put into the intestine, or we were giving bacteria that could replicate, and so we wouldn't actually know the dose that we were giving. And you can see here that as we bring down the dose, 10 to the 10, 10 to the 9, 10 to the 8, there is, between 10 to the 9 and 10 to the 8, a rapid drop-off in the number of plasma cells and also in the specific response measured out by the flow cytometric assay. This is actually quantitative histology. In other words, the threshold is set as being high. We respond to a very small proportion of the microbes that are in the intestine. And this response is an integral of the dose that we give. And the way that we addressed this was to look at giving either a series of doses, or to reduce the number of doses. And as you can see, as the number of doses goes down, so the response goes down. And I shall come to another slide to test memory rather more fundamentally in a moment. But in fact, it was cumulative with the number of doses, an integral of the number of doses. But if we separated out the doses, these 6 doses, they could be separated out at very different intervals, and actually it made no difference. So every 7 days, every 2 1/3 days, every day, always the same response at the end of the experiment compared with the germ-free control. Well, every day may be sufficient if we give 6 doses in order to maximally induce the system. So we decided on a rather more stringent experiment to either give a single dose of 3 times 10 to the 10 organisms over 24 hours, or to space that dose out over 3 doses and analyze it at day 24. And as you can see, in fact, the results were exactly the same dose. It made no difference splitting it up. And we could see within this no evidence of a prime boost effect looked at both with the IgA concentration in the intestine or the specific responses shown by the flow cytometric assay. So what happens if you leave it longer? And the answer is that the response is immensely persistent if the animal remains germ-free. So even following it out to 112 days, you can see that hardly any of this specific response, measured by specific flow cytometry, was lost. But if the animals were colonized, if one added in an altered Schädler flora, then the response became abrogated, or and I'm not going to show you this, or if we gavaged the mice with other organisms, we mono or dicolonized the mice, we also abrogated it. In other words, the response was persistent until another response took over. And this is a kind of non-prime boost intestinal memory. So just to summarize what I have talked about, I have tried to persuade you that there is a very high threshold for commensal bacteria to induce IgA. And I think this makes some sort of teleological sense, because it means that we are responding just to a tiny sample of the organisms that are present in the intestine. Remember, there is a very narrow difference between these immense concentrations of microbes in the lumen, or above the demilitarization zone of the inner layer of the mucus, and the rest of the body. And it is fundamentally important to us and to all animals not to become septic, to minimize the chances of an infection from these organisms, which they certainly can do in cases of, for example, patients on chemotherapy or post-radiotherapy. And so sampling a tiny proportion makes really quite good sense. The IgA includes specificities that are specific for the commensal surface. This is a one organism, two germ-free mucosal immunization, and there is a clear evidence of specificity. So it is not just a sort of non-specific combination of natural specificities. As in most immunology taught by viruses or bacteria, live organisms have a much more profound effect on the immune system than dead organisms. There is minimal immune memory, by which I mean there's minimal evidence of prime boost effect, although the response is very persistent until other responses are induced. And there is, in other words, persistence of one lamina propria specificity until the microbiota changes. And finally, in the earlier section, I tried to persuade you that the regulatory T-cell response is also important to preserve the subset of CD4 homeostasis in the lamina propria, irrespective of the other commensals or pseudo-aggressive commensals. Okay. So I'd like to thank you all for your attention, and also for our sponsors for the funding. This is actually Kathy McCoy's group and my group taken together, and I put up the names of those talented scientists that did the work, and I'd like to thank them as well.